Do you know your blood type? If you asked 10 people on the street that question, maybe 3 would answer correctly. The rest would guess, shrug, or say something like red. And honestly, that's not surprising. For most of us, blood type only comes up in emergencies, and by then, someone else is handling it. But here's what nobody tells you in that moment. The blood running through your veins right now isn't just yours. It carries a molecular signature so ancient it predates the human species entirely. Not by centuries, not by millennia, by 20 million years. We've spent over a century classifying blood into 4 tidy letters, A, B, AB, and O. We built entire medical systems around those letters. We saved millions of lives with that knowledge. And yet, we never actually understood where those letters came from, or why different humans carry different ones, until DNA changed everything. What researchers found buried inside our genetic code didn't just explain blood types. It rewrote the timeline of human evolution itself. It connected us to primates, to ancient mutations, to a biological mystery that science is still untangling today. This is the story of your blood, and it is far older than you. To understand why your blood type matters, you first have to zoom in, not to the vein, not even to the blood itself. You have to go all the way down to the surface of a single red blood cell and look at what's sitting on top of it. What you'd find there are tiny molecular structures called antigens. Think of them as flags. Each red blood cell carries these flags on its outer surface, and those flags are what your immune system uses to identify whether something belongs inside your body or doesn't. They are, in the most literal sense, your biological identity tag. Now, in the ABO system, there are 2 main flags in play, the A antigen and the B antigen. If your red blood cells wave the A flag, you are blood type A. If they wave the B flag, you are blood type B. If they wave both flags simultaneously, you are blood type AB, carrying both identities at once. And if your cells wave no flag at all, displaying nothing on that outer surface, you are blood type O. That last one sounds like a disadvantage, no identity, no flag. But as we'll see, it turns out to be one of the most powerful positions in the entire system. These antigens aren't proteins, which surprises most people. They're sugars, complex carbohydrate chains sitting on the cell surface, shaped just differently enough that your immune system can tell them apart. 4 letters, 2 sugars, and a system 20 million years in the making. Here's where it gets interesting. Because your body doesn't just display those flags passively, it responds to flags it doesn't recognize, and it responds hard. Think of it like a medieval kingdom. Your blood type is the flag flying above your castle walls. Your immune system is the army stationed inside. And that army has one standing order: attack anything waving a flag that isn't yours. If you are blood type A, your immune system never learned to tolerate the B flag, so it built antibodies against it, anti-B antibodies, circulating through your blood right now, ready to destroy anything carrying that foreign marker. If you are blood type B, the reverse is true. Your army carries anti-A antibodies, primed to attack the A flag on sight. Blood type AB plays no offense. Because it waves both flags, its immune system recognizes both as self. There are no antibodies against A, no antibodies against B. The army stands down entirely. And blood type O? It waves no flag, but it built the largest army of all. Anti-A antibodies and anti-B antibodies, both, circulating simultaneously, ready to attack either foreign marker that enters the bloodstream. This is why blood transfusions can kill. Send type A blood into a type O recipient and those anti-A antibodies mobilize instantly. The immune system doesn't see medicine. It sees invasion, and it responds with everything it has: clotting, organ stress, systemic shock. Your blood isn't just a delivery system, it's a fortress. So if blood types are this incompatible, how does emergency medicine even function? Someone arrives unconscious after a car accident. There's no time for testing, no medical history on file. The clock is already running. What do? You reach for type O negative. O negative is what medicine calls the universal donor. And the reason comes directly from everything we just covered. Type O cells wave no flag, no A antigen, no B antigen, no marker for any immune system to recognize as foreign. Whether the recipient is type A, type B, type AB, or type O, their antibodies have nothing to attack. The blood slips through without triggering a response. Add the negative component, meaning the rhesus factor antigen is also absent, which we'll get to shortly, and you have blood that virtually any human body will accept without protest. On the opposite end of the spectrum sits type AB positive, no anti-A antibodies, no anti-B antibodies, and full tolerance for the rhesus factor. AB positive can receive blood from every single type in the ABO system without rejection. Medicine calls this the universal acceptor, a body that simply will not turn donated blood away. These two positions, universal donor and universal acceptor, sit at opposite poles of the same system. One gives to everyone, one accepts from everyone. And between them, they represent the outer edges of what human blood compatibility looks like. In an emergency, these two blood types carry more weight than almost anything else in the room. For the first half of the 20th century, doctors believed the ABO system was the whole story. 4 blood types, a clear compatibility chart, and a set of rules that finally made transfusions survivable. It felt complete. It wasn't. In 1937, 2 scientists named Carl Landsteiner and Alexander Wiener were running experiments on rhesus monkeys when they noticed something unexpected. When they injected rhesus monkey blood into rabbits and guinea pigs, the animals produced a new antibody, one that reacted not just to monkey blood, but to human blood as well. Specifically, to the blood of about 85% of humans tested. That meant 85% of people carried an additional antigen on their red blood cells that nobody had accounted for. They called it the rhesus factor, the D antigen. And just like that, 4 blood types became 8. If you carry the D antigen, you are rhesus positive. If you don't, you are rhesus negative. And the rules that apply are unforgiving. Rhesus negative individuals don't naturally carry anti-D antibodies, but expose them to D positive blood even once, and their immune system learns. It builds those antibodies and remembers. Every subsequent exposure triggers a stronger attack. This has life or death consequences in pregnancy. A rhesus negative mother carrying a rhesus positive baby can develop antibodies that cross the placenta and attack the infant's blood cells directly. One extra flag, discovered in a monkey. And it fundamentally changed how medicine approaches blood and birth forever. To appreciate how far we've come, you have to go back to a time when nobody understood any of this. No antigens, no antibodies, no compatibility charts, just curiosity, desperation, and a staggering willingness to experiment on living human beings. For over 1,000 years, medicine operated on a theory proposed by a Greek physician named Galen around 200 CE. Blood, he argued, was continuously produced by the liver from digested food, consumed by the body, and never circulated. It was a static system, a one-way pipeline. And that idea went essentially unchallenged for nearly 14 centuries. It wasn't until 1628 that a British physician named William Harvey published the work that changed everything, demonstrating that blood actually circulated continuously through the body, pumped by the heart in a closed loop. It was one of the most important medical discoveries in history, and it immediately made people want to start experimenting. By 1665, an English physician named Richard Lower successfully kept a dog alive by transfusing it with blood from another dog. The scientific community was electrified. If it worked in dogs, surely it would work in humans. It did not work in humans. Within 2 years, doctors began attempting xenotransfusions, transferring animal blood, primarily from sheep, directly into human patients. The results were fatal. Patients convulsed, their urine turned black, and they died. Nobody understood why. They had no framework for incompatibility because they had no concept that blood had types at all. The knowledge would take another 230 years to arrive. By the turn of the 20th century, blood transfusions had a reputation problem. They worked sometimes and killed people other times, and nobody could predict which outcome was coming. The medical establishment had largely given up on them. Too unpredictable, too dangerous, too many funerals. Then a quiet Austrian physician named Carl Landsteiner decided to find out why. In 1900 and 1901, working in a small laboratory in Vienna, Landsteiner ran a series of methodical experiments. He collected blood samples from himself and 5 colleagues, separated the red blood cells from the serum, and began mixing them in every possible combination. What he observed was systematic and repeatable. Some combinations clumped together, the cells agglutinated, clotting into visible masses. Others mixed freely with no reaction at all. The pattern was not random. It was governed by something intrinsic to each person's blood. Landsteiner identified three initial groups, A, B, and C, which was later renamed O. A fourth group, AB, was identified the following year by two of his students. The ABO blood group system was born, and with it, the entire modern framework for safe transfusion. Landsteiner received the Nobel Prize in Physiology or Medicine in 1930, nearly 3 decades after the discovery. In the years between, his classification system had already transformed battlefield medicine, surgical survival rates, and emergency care across the world. One man, 6 blood samples, a laboratory bench in Vienna, and a discovery that has since saved hundreds of millions of lives. Here is something most people never learn in a doctor's office. The ABO system, the 4 letters, the compatibility chart, the universal donor and acceptor, is not the whole picture. It is not even close to the whole picture. Scientists have now identified over 40 distinct blood group systems recognized by the International Society of Blood Transfusion. More than 300 individual blood group antigens have been cataloged. The ABO system is simply the most immunogenic, meaning it provokes the strongest and most immediate immune response, which is why it became the foundation of transfusion medicine. But the others are real. They matter, and in certain situations, they can be just as dangerous. Take the Duffy blood group. Discovered in 1950, this system involves antigens on the surface of red blood cells that also happen to serve as entry points for the malaria parasite, Plasmodium vivax. People who lack the Duffy antigen, a trait found in a significant portion of people with West African ancestry, carry a natural resistance to that specific strain of malaria. Their cells simply don't give the parasite a door to walk through. Then there is the H blood type, sometimes called the Bombay phenotype. Extraordinarily rare, appearing in roughly 1 in 10,000 people in India, and far fewer elsewhere, these individuals lack the foundational antigen that all ABO blood types are built upon. They can only safely receive blood from others with the same rare type. Blood, it turns out, is not 4 things. It is hundreds. Before I show you the last part, if you want to look at your own results and see where ABO blood types fit into your bloodline, I put a full 3-volume PDF bundle over at the OriginDNA website. It shows you how to read your raw data and find matches beyond what Ancestry or 23andMe tell you. Link is in the description. But stay right here because what comes next is the part most people completely miss. Here is where the story stops being about medicine and starts being about something much deeper. For most of the 20th century, scientists assumed the ABO blood group system was a relatively recent development in human evolution, something that emerged in our species, shaped by our specific biology and environment. A human invention written in molecular code. Then DNA sequencing arrived, and it told a completely different story. When researchers began mapping the genetic variants responsible for ABO blood types across species, they found them far outside the human family tree. Chimpanzees carry A and O blood types. Gorillas carry primarily B. Gibbons carry A, B, and AB. The same antigens, the same molecular flags, appearing across primate lineages separated by millions of years of independent evolution. The implications were staggering. For all of these species to share the same blood type variants, those variants couldn't have evolved separately in each lineage. They had to have been inherited from a common ancestor, a single ancestral population that lived and diversified long before modern humans existed. Genetic dating pushed that origin point back approximately 20 million years, before Homo sapiens, before Homo erectus, before the entire genus Homo existed at all. The ABO system was already ancient by the time our earliest recognizable ancestors appeared on the evolutionary timeline. Your blood type was not created by your species. It was inherited by it. Passed down across 20 million years of primate evolution, through extinctions and migrations and continental drift, arriving intact inside your veins today. So the ABO system is 20 million years old. It survived the entire arc of primate evolution, crossed species boundaries, and arrived in modern humans fully intact. Which raises the most obvious question of all, and the one science still cannot fully answer. Why? Why did natural selection preserve 3 distinct blood type variants across 20 million years and dozens of primate species? Evolution is not sentimental. It does not maintain complexity without reason. If A, B, and O had no functional difference, if carrying one versus another offered no survival advantage, the system would have collapsed into a single type long ago. The fact that all 3 persisted suggests they each offered something. But what exactly remains genuinely contested? The leading theory involves disease resistance. Different blood types appear to confer different levels of susceptibility to specific pathogens. Type O individuals show some evidence of increased resistance to severe malaria caused by Plasmodium falciparum, the deadliest strain, but they appear more vulnerable to certain cholera strains. Type A has shown associations with higher susceptibility to smallpox historically. Type B carries different risk profiles again. The theory, broadly, is that no single blood type is universally superior. Each offers protection against certain pathogens while carrying vulnerability to others. Maintaining diversity across a population means the species as a whole is never completely wiped out by a single disease. It is evolution's way of hedging its bets, spreading risk across a population so that no single outbreak can eliminate everyone at once. A 20 million year insurance policy written in sugar molecules on the surface of your blood cells. Blood types are not evenly distributed across the human population, and that uneven distribution tells a story that DNA is only now helping us fully read. Type O is the most common blood type globally, carried by roughly 44% of the world's population. It dominates in indigenous populations across the Americas, where in some communities it approaches 100% frequency. Type A clusters heavily across Europe and in certain Central Asian populations. Type B appears at higher frequencies across parts of Asia and the Indian subcontinent. Type AB remains the rarest everywhere, never exceeding more than a few percent of any regional population. These distributions are not random. They reflect ancient migration patterns, population bottlenecks, and thousands of years of localized disease pressure shaping which blood types survived in which environments. Your blood type is, in a very real sense, a geographical record of where your ancestors lived and what they survived. Modern DNA testing is beginning to bridge that gap, connecting blood type genetics to broader ancestry markers, helping people understand not just their ABO status, but the ancient population movements embedded in their genome alongside it. And then there is Japan, where blood type became something else entirely. Since the early 20th century, Japanese culture has linked blood type to personality, a system called Ketsueki Gata. Type A is considered organized and anxious, Type B, independent and selfish, Type O, confident and insensitive, Type AB, rational but indecisive. In 2011, a Japanese government minister publicly blamed his volatile behavior on being Type B after a political scandal forced his resignation. Science does not support the personality connection, but 20 million years of evolution, that part is very real. 20 million years ago, a primate carrying a specific molecular flag on its red blood cells passed that flag to its descendants. Those descendants diversified, migrated, and eventually became every primate species alive today, including us. The flag survived all of it. Ice ages, extinctions, continental separations, and the entire sweep of human history. It arrived in your bloodstream this morning, as reliably as it existed before our species had a name. Your blood type is not a medical footnote. It is an evolutionary document, a molecular record of survival strategies older than humanity itself, written in sugar chains on the surface of cells you never think about until something goes wrong. And we are only beginning to read it properly. DNA is unlocking connections between blood type, ancestry, disease resistance, and ancient migration that the ABO chart on a hospital wall never hinted at. The 4 letters are just the beginning of a much longer story. If you want to know your blood type, donate blood, ask your doctor during routine blood work, or pick up a simple home test. It takes minutes. The answer it gives you is 20 million years old. Most people look at their DNA percentages and stop there, missing the real story. Over at the OriginDNA, I built a 3-volume PDF guide bundle to fix that. It shows you exactly how to analyze your raw data, break down matches, and trace your deepest ancestral roots. The link is right at the top of the description. Click it, grab your copy, and I'll see you in the next video.